Ken, welcome, Ken.
Hi.
Hello. Yeah.
Hi.
Okay.
Nice to see you. Thank you. So I will share the screen first. Please let me know if that works well with you. Start screen share. Is it now? Can you see now my screen?
Yes.
Yeah.
Okay. So the talk I will show you. Oh, wait a moment, because I have a problem now. That what is here next? Okay. From the gametes to the blastocyst, changes in cell surface morphology. You know, I am a morphologist. I am a doctor basically, and I understand only morphology. Molecules I don't understand, so it's the only chance for me to do some research to see the shape of the things. And I'm passionate with microscopy, scanning electron microscopy. And here I will start the story on the human life from the beginning, in a way. That here you see an ovum, a human egg, which is at the time of ovulation, is surrounded by the cumulus cells called corona radiata. These cells actually they feed. Here is a graphic. These cells they communicate through projections and meet projections from the ovum, and communicate through gap junctions, and it is the way that the egg is loaded by all these substances, nutrients, and cell machinery components in order to grow and to be able to sustain the new life. And I think actually that the perfect round shape of the egg is due to this action of at least 5,000 tiny cells, normal cells, but the ovum is big, that they act all over and they act equally, so it gets this round shape. It's just a thought. Now, what happens a little bit before ovulation, because the human choreotrophic human gonadotrophin is secreted, the LH, sorry, the luteinotropin hormone, and then this projection I showed you before, they withdraw both sides and they leave empty spaces. Here, by the way, is the egg cell. It is the Zona pellucida by blue, and inside is the ovum itself. And here is the first polar body. And no one knows how the sperm really makes its way through the cumulus corona layer and the thick and elastic Zona pellucida in order to enter and fertilize the egg, because to fertilize it has to melt with the oolema, we call the membrane of the ovum. But I have, and people think that it makes its way between these corona radiata cells, and then it digs a channel inside the Zona pellucida to meet the ovum. But I will show you evidence that the sperm actually goes through the corona cells, not between, through the cells, and it uses one of these pores that is left by the empty space is left by the mutual withdrawal of the projections to meet the. So next slide now. Yes, this is now corona cells. This is the Zona pellucida in yellow, and these are the projections. They were deep inside, and now they withdraw. This is an ovum that we partially remove the corona cells to expose the zona pellucida here. You see, yeah. Something is wrong. I'm sorry, I will start again because I don't have the right menu here. I will go like this. I don't know what happens, and by scrolling it doesn't move ahead. I have to use this next. Anyway, and this is a detail of the previous image of here somewhere, and you see in detail how the zona pellucida is made from 3-4 layers of rings. You know, they look like rings here, and in this slide, I put together in the same magnification a mature egg on the bottom and the immature egg. And you see in the immature egg that these pores are very little because it didn't have the time to expand, you know. And this is the mature egg with expanded pores, and this ring-shaped structures is very important for fertilization, I think. Now you see another SEM image. It is full of little pores here. Yeah. Let's talk about the sperm a little bit, because it is, we tend to ignore the male factor, you know. And this is sperm traveling its way through the fallopian tube to meet the egg. This is a perfect wave if you follow this. There was another curve here, but the agency cut it because it found it confusing. But actually, it makes 7 curves of an harmonic wave. It's very difficult to get the sperm so active to kill it so active. Usually it gets like a spaghetti, like this one, you know, because it has the time to do some movements and before dying. This is now the fallopian tube here, and I have put an egg here to show the actual size of the. I put it, we didn't find in the woman this. And this is the fallopian tube, and there is here sperm, if you see inside here. There is sperm. It's very rich, this visually. It's like the bottom of the sea, you know, like the seafloor. This is sperm, again, in the fallopian tube. Now this is the egg. At ovulation, it has, as I mentioned before, it has the corona cells around and the zona pellucida in yellow, you see, inside. And the sperm has to pass first these cumulus cells. The way it passes is a mystery. But if you see this slide, this sperm has lost the acrosome. The acrosome is the cap here, which should be lost in contact with the zona pellucida. This sperm prematurely lost the acrosome. It underwent the acrosome reaction and the hyperactivation, which is needed to pass through the zona. But here it's prematurely happened. You can tell it is hyperactivated by this shape of the tail. And actually it is stuck on the, here on the one corona cells. It will not go further. Other cells, other sperm that goes further, you see here, it looks like it penetrates the corona cells. And such figures that have been interpreted like phagocytosis of sperm by corona cells, which I don't think it's true, this, because the sperm it's not damaged, it looks very active, You see these figures, it looks like it enters here, the corona cell, one another here. And if you do a transmission electron, you see here that it looks like it swims freely within the cytoplasm of a corona cell. There is a halo around, there is no evidence of phagocytosis. And actually the sperm is used to be in the cytoplasm of other cells because it grows in the Sertoli tubes in the how do you call the, now my mind is not going. Anyway, inside the spermatogenesis is happened within the cytoplasm of Sertoli cells. So the sperm is used to be inside somewhere else, you know. And when it is free, it gets very anxious to get somewhere in, you know. But let's talk about the next layer, which is the zona pellucida. It's very important for the selection of the sperm also. And also it blocks polyspermy in the way I will show you soon. And the zona pellucida has to shed off before implantation. Now, no one knows how the sperm really penetrates this elastic coat, the zona pellucida. And they have been developed two hypotheses, the mechanical, that is primarily mechanical forces, and the enzymatic, that the primary role is enzymatic reactions. This I got from Yanagimasi, who was, I think, the best so far, Japanese, but living in Hawaii, I think, to study fertilization. And now I have a very important physical question that, as we know, the sperm strives to enter the egg. So why it doesn't turn away? Because I know from physics that any moving object, if it encounters resistance, it will turn away somehow. It cannot stay exactly opposite to somewhere. It will turn, it will take an angle and then escape. So what is this that holds the sperm firmly there, and it can only move inwards? It cannot, and I think the answer is not enzymatic, or it is, the answer is mechanical, because if you see here all these rings in the egg here, sorry, the image is not so good, and we see the sperm that actually enters the zona pellucida through these rings, these pores. If you see these images here, so must be something like here I put together that once the sperm enters the outer circle, it is entrapped there and can only move inwards. It cannot turn around. So it is like a mechanical factor that guides the sperm inside. And I like to compare it with the sex, you know, sex in the microcosm. Because if you see, if you see how similar is the sperm with the penis, it is remarkable, the similarity, you know, and even the head is like the head of the penis, also the acrosome is like the post, we say here, the cover of the, and also the rings of the egg are, the vagina has rings here. So it's like sex in the microscope. The nature repeats itself. And at that time of fertilization, the egg is in the fallopian tube. So that's what we believe, at least, because no one really knows. After the one sperm enters in the egg, immediately there is a big reaction of the egg. Waves of calcium are running all the cytoplasm of the egg, and there is the release of the so-called cortical granules, small cysts with enzymes below the surface of the egg. And this has proteolytic enzymes that they change immediately the zona pellucida from fenestrated, as you see here on the left, to a compact one, as you see on the right. And actually, when I published this, the referee told me that, how do you know it's not an artifact, this, for example, and it got compact because of... and I say to him that if you notice, the outer rings, they remain intact here. If a force from outside has made the zona compact, it would affect first the outside rings. So it comes from inside. So I believe that these elastic hoops, as we call the rings, may act as mechanical receptors for sperm. This important function of the zona pellucida now is completely bypassed by the sperm microinjection. You know, that happens in IVF, and it happens now in 95-97% of the cases. They do immediately high technology, even if the husband has a good sperm. You know, this is not good. Anyway. And this is, we did the work with one group from France, and they had a population with some cyclin, which is a cell cycle enzymes, doesn't work, and the meiosis doesn't work, and 4 spermatids remain joined, and they are completely infertile, these guys. So, to carry on now to the embryo, I use the mouse model to describe what happens. After the second and third cleavage, that renders 8 cells in the mouse, then a very important function happens, which is called compaction. So in compaction, there is segregation of the cells to the inner and the outer. The embryo does not starts the first sign to behave like an organism and not like separate cells. And after compaction and the mitosis of the cells continuing and the cell numbers increases, and there is fluid through apicobasal fluid transport, fluid accumulated inside the morula that pushes the inner cells to one side, and they will form the inner cell mass, which will form the embryo itself, and outside remains the outer cells, the trophoblast, which will generate the extra embryonic tissues. We don't know, we did a study to see what is the timing of compaction in the human embryo, because there was no much data about it. It seems to vary between various species. As you see here, the bovine is later, in the pig is even later. And we started from unfertilized, this is an unfertilized egg, but I removed the zona pellucida, and you can see the inside, the oolemma. It's called oolemma or vitelline, maybe you're more familiar with this vitelline membrane. And this is the first polar body here. And we see there is a lot of microvilli here. You see, it's plenty of microvilli, and some long ones at the area of the first polar body. Below the polar body lies the spindle. Yeah, which because the egg is arrested at this stage of, and this is the fusion of the sperm with the egg. At one point it fuses. We have cut this point, but at the moment we had low resolution machines and we could not. I could not take a better picture. But immediately after fertilization, the number of these microvilli decreases dramatically. And there is a theory that this microvilli works like a reservoir of membrane components that later are used to form the pronucleates and other structures inside the cells that they need for to sustain growth. Now we are at the two-cell stage. This you never see unless the egg, the embryo gets arrested. It is very fast stage, the second cell, the two-cell stage. Then it is the four-cell stage. Nice embryo with equal blastomers. You see the Zona pellucida here around, quite thick. Here I open the Zona pellucida. This is electron microscope now. And you can see the blastomers, we call blastomers inside this. It's the same in axolotl, I think. And this is four-cell stage. Still, there is no signs of compaction. This is a poor quality embryo with various things on the top, like spreading one cell to the other. We don't know what is the significance of these functions. This is a 8-cell stage, even more at the day 3. This is not 8, this is 10, 12 cells. Day 3, still no compaction, and the blastomers are held together by long microvilli connecting the two blastomers. Still, the embryo, as I told you, at this stage, behave not like a single organism, but they are just an aggregation of cells. They don't communicate much to each other, and that gives the opportunity to remove one blastomer and make genetic diagnosis to the embryo. You see here is the opening in the Zona pellucida. It is at the time we did it with acid thyroid, now we do with laser. And this is an embryo which I put on the top of a needle, on the tip of the needle, to actually show the size of the embryo to people that they are not familiar, with microns and all this. And they can get an idea of how little is the embryo. And this is important for people working on ethical aspects, at least to know that the embryo is not anything with eyes or legs, or it's just an aggregation of cells. And actually the term that was valid for many years was fertilized ovum, not embryo. The term embryo, and the American term pre-embryo is more correct. The term embryo emerged after in vitro fertilization because it was too much to say unfertilized egg, fertilized, and they say embryo, and it's more important, Yeah, but that created a lot of hassle in the ethical point, And here is the same embryo, and look, there is a sperm here engulfed by one extra sperm, because in vitro you put many sperm and maybe one passes, but it didn't incorporate. But this can create a big mess in genetic diagnosis if you remove this blastomer for genetic analysis and there is a sperm on it, you will get funny results. Now look, this embryo is one stage further and starts to compact. So the blastomers flatten to each other, but they flatten also on the apical sides, They flatten to each other, but also there. And it is the first sign of compaction. This is more compacted. Embryo, morula stage, day 4. And if you segregate the blastomers, you see that there is now differentiation of the cell surface. At the points of contact, there is no more long microvilli. The microvilli disappear, and there are areas of contact with some bubbles here. Yeah, and this is more compacted morula. And we kept the time, and this is a bad embryo with a lot of how do you call these bubbles, like also this is a bad embryo, bad quality for IVF. And it is interesting that it fits one blastomer to the other like this, like a toy, you see. And if you segregate, you see it's like a boy's toy that fit together to one to the other. We don't know what is the significance of this phenomenon. It's difficult to investigate. So from this study, we concluded that the human embryo undergoes a strong compaction at the 10 to 16 cell stage on the day 4 post-insemination in vitro. In vivo, the knowledge is so poor that you cannot tell anything. Which is a pity now that we need a lot more information about what happens in vivo, because we interfere and we do in vitro fertilization. Now is banned the research on human embryos. Before it was allowed, and now it's banned. Strange, eh? Yeah. And we did the cover of this for because the photos are appealing, and I don't know if my work was so significant, but they did the cover anyway. So now we go further, and now the egg is in the, not anymore in the fallopian tube, it is in the uterus. This is the uterine epithelium. This is an egg. This is an embryo, actually, because it's still enclosed on the zona pellucida, but inside must be a morula, as I showed you before. And now let's talk about blastulation. I already mentioned how it happens with the inner cells, the outer cells, and the transport of fluid to push the inner cell mass to one side. And this zona pellucida has to be shed before implantation. And how this hatching happens. Look, this is a human embryo. It is day 5, and it started to hatch. You can see this part of the trophoblast protruding from the zona pellucida. This is the same embryo on the tip of the pin for electron microscopy studies. And here is the point of hatching. It's very interesting that it is very, very little. If you see how small is this opening, it's very small. Eh? It's like an atomic bomb. How do you call this mushroom of the atomic bomb? It's a very dramatic moment in the life of embryo. And it needs to be a strong embryo to undergo hatching. And then the first, from the hatching point, which was first a little circle, now the embryo managed to make a long slit from there, and already a big part of the embryo is getting out. This is now with laser. We do this with laser for blastocyst biopsy. We never let the poor embryos in peace, you know. And this is the holding pipette, as we call, through suction. It holds the embryo on the spot, and here is the laser, the action of the laser, that if you see the same embryo here, it's the same embryo in electron microscopy, and some cells are really fried here through the action of laser. You can tell from the membranes how they are, but there is some protein around that creates this mesh. And this is now assisted hatching, we call. You open a hole in the embryo, in the zona pellucida, in order to facilitate hatching. I don't know if that really helps implantation, you know, because maybe if the embryo is vital, it will hatch by itself. But anyway, people do it. Yeah. This is now more advanced hatching, figure of 8, we call it, here the embryo, here with colors from my wife. My wife is designer and she's helped me in this work. This is the embryo and this is the zona pellucida. So it could not stay longer here. It wants to expand, and this is now look, now I'll show you crack opened embryos to expose the inner cell mass. And this is an embryo that looks from outside, the trophoblast looks of good quality because there is here a lot of microvilli. There is some specifications here which I will show you later that they are important for signaling towards the uterus. But if you crack this, exactly this embryo, you see inside the inner cell mass is degenerate. You only see fragments and bubbles, blebs of cells. And so it can be that the inner cell mass doesn't go well, but the trophoblast is still alive, and that gives pregnancies, but the so-called unembryonic pregnancies, where you get a positive pregnancy test and a sac, but no embryo. It is very embarrassing situation and sometimes needs curettage. Now this is a good embryo, day 5, enclosed in the zona. We open it to expose the inner cell mass. I will show you here more details. This is the inner cell mass, and this here I will show you better in color, which is the inner cell mass is here in red. And you see there are some cell debris here, some cellular death, but not so much, not like the previous one. There is always some dead cells. And here is an interesting point at the junction between the trophoblast and the embryoblast and the inner cell mass. There is one cell that has radiated many projections, touching many other cells, and the other cells are also radiate projections. So it seems that this mass communicates very strongly, and maybe this is the organizer, as we call the organizer, that organizes the inner cell mass, that already from day 5 has some shape, maybe has up and down, left and right. And now another embryo with, what is this? Yes, another inner cell mass, good inner cell mass here you see in light microscopy. It started to hatch on day 5 also. I put it first here, and the inner cell mass goes down because of physical laws. It is more mass at this point, so the needle attracts more the side of the inner cell mass than the other, and it jumps on the needle because, okay, it jumps by itself. I don't put it myself there. I could never put one thing that is 1/10 of millimeter to catch it and put it there. It does by itself, and if you open it, there is a lot of protein inside, which you need to brush it out, and then you see the inner cell mass here. Yeah, and this is the point of hatching from the inside. This is the so-called blastocell. It is the cavity. Okay. This embryo had two inner cells. One is here, and there is a second mass here. You see, a little bit black, maybe not so vital. And after I opened, there were two inner cells, one this, and the other this one. Already twins, you know. And this is a hatched embryo, but you think by this picture that it's a good embryo with many cells. It is a bit black though. And if you do scanning, electron microscopy, you see that this embryo is no good because the cells are in bad shape. You see the plasma membranes, they are fragmented. And now finally I will show you the best embryo I ever had, which is a big blastocyst day 6 with expanded blastocyst. Now it collapsed a little bit, it's not perfect round. This is the light microscopy, inner cell mass here, and now on the way for microscopy on the tip of the pin. And here you see many, on the plasma membrane, there are many here, many, I don't know what is happening. There are many like ruffles, like some things that it's difficult to tell what they are doing. And there is this was studied years ago by in the mouse. This is an old publication I found, and they comment on these ruffles, but they say they resemble like cancer cells and so on. And I could not understand what these ruffles are doing until I saw this. Look, it was the only one here. If you see here, this is what I will show you in more detail. This one here. First, I thought it was a rubbish. I didn't pay much attention, but if you see in this image here, this and this and this, maybe the same structures at different stages. It looks like a huge cyst, how do you call it? A huge, yes, cyst is ready to exteriorate and to secrete it in the environment. And this contains a lot of granules inside. There is some remnants of plasma membrane here. And this must be a huge secretory granule. And this is the reabsorption now of the cap remains here and then gets reabsorbed like here, you see. This may be the same like this, but it released its content and then the plasma membrane gets reabsorbed. And here you see different stages. I think this is a very important point in the life of embryo, because it has to make felt its presence in the womb, and to send molecules to signal to the womb and start implantation. So that's it in a way, and yeah, this you see that the area overlying the inner cell mass is different, has some long microvilli here, and this is the area we believe that the implantation starts from. It implants first, and it behaves very aggressively. So this is, I would like to thank my wife for contributing, and also these German guys, they did a fantastic machine allowing me to get very high resolution pictures, and very long, of course, yeah. So that's all what I wanted to show, and I have also a series of these blastocysts that this is on the top of the endometrium. It attached, but I think it is a non-specific attachment through dead cells. I don't think it taught us many things more than a nice photo, Yeah. So that's it. I think I finished now. At this moment, I have some more photos. One blastocyst that I can show you all around, but yeah, this is interesting for me also to ask questions from you, to ask you questions about how can I visualize all the embryo around, with minimum of photos, because they take a long time. Okay.
Yorgos, you left 8. I see you have 8 more slides there.
There are more, yeah. The more slides are look. It is this series of the.
Oh yeah, show that. Show the series.
The series. The series is the same embryo in different aspects, and you need to put landmarks. For example, this one here. Okay, you will see it's here, and the second photo is now here. The same thing.
This is by rotating the needle, right?
By rotating, you can tell from the needle. Also, it has these two here, and this is the cyst I showed you before. It's here, and now the cyst is here, and this is here. Now we turn clockwise, and the cyst is here, and this point from here gets to here, and then another side here. Now, yeah, the cyst. This is the cyst, and from upper. So I turn around as many times as to have the whole thing, and that's why I put on the tip of the pin, so I have maximal area to be viewed, because I cannot find any other method through we were talking with Dick that maybe through magnetic forces can stay on the air, but it sounds difficult for the moment, and for my knowledge of physics looks impossible. So that's it, Dick. I have many more slides, but I didn't select it because it will be redundant, But if you are interested in any specific points, I can expand with slides.
How much of these ideas have you published?
Sorry?
Have you published, for example, your observations on the hatching?
No. I only published this compaction, and then I don't publish because, now you have even to pay to publish, you know. Why shall I pay to publish? They should pay me somebody for doing all this work I found by myself, because I love research, but I don't want to pay, you know.
Yeah.
Yeah, I think it's not justice, this policy, you know. But I have the data. Sometime maybe I will publish. Yeah. But already what I'm doing now, it's a sort of publication, you know. Maybe finally YouTube will be have more impact factor than big journals. Yeah. Yeah. So you have glasses, eh? No, they don't work. These glasses, they need special screens and softwares that they work with polarized light. I have slides, but they are red-blue, you know. A bit primitive way, but it's the only way I can afford.
Yeah, the old I'm not sure where we get any red-blue glasses now.
Yeah, they seem to be like relics now.
Yeah, yes, they are. Everything is polarized now.
Yeah, everything is but you need special software and special screens that they emit polarized light, you know. It's expensive equipment, this.
Yeah.
I would love to do this, because like this you can have many colors also.
Well, there are two tricks that are available that don't require glasses. One is to present a stereo pair, and then it's a difficult skill. But if you cross your eyes the right way, you can see it in 3 dimensions.
Yeah. Actually, okay. This was yeah.
Okay. The distance between the pictures has to be small enough so it's the same as the distance between your pupils. But the other way, which you can do here with the needle, is there is a psychological effect where if you see a movie of a rotating object, then you see it in 3 dimensions as it moves. As soon as the movie stops, it becomes flat again.
Yeah.
So it would be possible.
But
Bradley, he does need help with something which we've been discussing. Yorgos can rotate the embryos nicely on the needle, okay? But there's a little bit of jitter, you know, because we're dealing with very small dimensions here. And he needs what seems to me relatively simple software to bring the images back to a common axis of rotation so that they make a smooth movie as they rotate.
Maybe I can show now the series of... so Bradley will have a direct view. Let me find the file, just a second. Yeah.
Now see, and the other thing is if we did that, it should be possible. I know there's some literature on stereo microscopy from SEMs for producing a 3-dimensional picture from a 3-dimensional data set from stereo views. He's got rotation all the way around, so it should be possible to extend that software to make a full 3D reconstruction of the surface. Okay? So I think that would be an excellent place to start collaborating with Yorgos, is to take these embryos rotate at the tip of the pin and start producing 3-dimensional structures from them. Okay.
I think just to add, I think you can actually do if you got 2 stereoscopic images, then you can actually do it like Google, what is it called, the Google Cardboard. So you can actually use use the phone or something like that to actually see stereoscopic images, because you're basically projecting two images, the stereoscopic images, and you see it through a goggles, kind of using your phone to see a goggle. That's what the cardboard Google does. So you can actually do a very simple one too. So without
that requires the Google glasses, or
no, it's quite interesting. You're using your phone, your Android phone. You put it into a cardboard sort of structure. It's a cardboard, and then you put it in there, because you actually split the two images.
Oh yes,
so you can actually see video, stereoscopic videos.
Yeah, there's a fellow. His name's Richard Feldman. He had, in fact, I used to own one of those boxes, but it was larger, and he made stereo slides of models of proteins. And the box would allow you to superimpose the two images in stereo.
Yeah. So, but now I think with Google, I think it's got some API, so you can actually do it more easily. You can get those images. I think you can do it. I haven't done it, but it's just because of the development.
You know what this phone accessory is called? It's called.
Yeah. Yeah.
I think it's probably not lens. It's probably just prisms. Yeah, because
I don't think it doesn't even use a prism. I think it's just a division. It's just a physical division of two of the screen into two. So you're actually seeing with the same screen is projecting two images but split, and then you see it from the cardboard, which is just a physical slide between two images. So it's very simple and easy to do, and it's very cheap. I think it's about, you can get the cardboard for about $30 or less. The Google Cardboard, and it's got it comes with software, and the software is probably free and downloadable and things like that. But you need to kind of combine the images in such a way that you can do it, I think.
So basically it's sending one image to each.
Yeah,
so one image to each eye.
Yeah, basically it's stitching two images into one image and projecting it on the screen, the split screen, and then you use the cardboard just to physically stop those, and then you just put it on your head and you can see it. So that is probably the cheapest and simplest to do it without costing a lot of money.
Interesting. Now I have a question that maybe Ken knows better. That imagine I want to videoscope all the one embryo, like the last I show you, all the way around, like 360 degrees. Do I need to take all the angles like one by one, or I can take some and then a computer can put the rest? And how many? I guess that it will depend on the relief also of the surface. How deep it is.
I don't know whether I'm not so sure you can easily. At the moment you can't. I think you need to have you need to create a. I think to do it manually is very difficult. You can do it manually, and you somehow have to use the software to kind of rotate and look at it. You probably, I don't know how easy it is to do the experiment and to see it, because I think the electronic microscope takes a long time, and basically you need to have a rig such that you can rotate it. Yeah, at the moment you're rotating it by hand, by the look of it, and I think it is quite difficult. Then you need to basically as what Dick was saying, that you need to find the corresponding points, which you know.
You know what? Susan probably has a solution. Susan, could it be possible to put a tiny motor inside the vacuum, inside the SEM to rotate that tin?
It is possible, and there is such machines that they do.
Susan is working with very small motors to build this. She's built a stage for rotating the axolotl embryos.
Mm-hmm.
There are small motors available. What size? That's the question.
The size?
Microns. It's very small.
No, but how big a motor can you get inside your SEM?
Yeah, you can put things inside the SEM, and there are special motors like micro tools. I can show you right now on the web. There is a German and they give I don't know how little it goes, but must be like 1/100th of a degree the rotation each time you press. With very high.
That's overkill.
Yeah, it's very, very accurate, no drifting, but they cost a lot, maybe 5,000-6,000 to one of these.
Oh yeah, well that's Susan. How much do your motors cost?
I've got them for $50.
$50.
Yes.
The other thing I was wondering whether you can do it manually and you put a artificial marker somewhere. And then you rotate it. Then, therefore, you can use software to automatically translate or calculate how what you have moved, such that you don't need to buy that. I don't know. I'm just guessing.
Yeah.
Whether you can do that. So you basically have a specific point or a couple of points that you can actually triangulate using software that it is there. And then you manually move around, you manually rotate it, and then therefore you may be able to reconstruct the whole thing in a movie. Yeah, in the movie. How many? I don't know. I mean, Dick may know actually more, but I mean, normally you need 30 frames per second, so you need a lot of frames, right? And you can,
you mean in terms of smooth rotation?
Yeah.
Just showing movies of people is about 30 frames a second, but it depends, of course, on how fast you're rotating. I think the question really is the question of how much detail is lost if you make the angle too big.
Yeah, exactly. How much? Somebody told me that he's doing. There is a German guy doing. He called nano flight. If you see, he has special stages, very expensive, that they rotate. They do all sorts of movements. And he told me that if the rotating object is rotates on its axis, central, then you can move 1 degree per frame. If it is eccentric, the rotation, then you have to go much less in order to achieve a smooth rotation.
Susan, do you have your stage handy?
Yeah.
Could you show everybody what you've got there? Because this is made with small motors.
It's called a nano motor, but can you see that? It's right here. So how big is it compared to my finger? Well, it's fairly. You can get smaller motors than this, but this is a nano stepper. I don't know if you can see that.
Hold it in the center of the camera. You're off the edge center of the camera.
Okay.
There you go. That's better.
Yeah. So this one here.
Now this is a stage for holding the axolotl embryo, and it has 2 different motors in it for 2 different rotations. And the whole cost was what, about $400?
I don't know. The big motor was fast and more expensive. It's about 70. It costs more to machine it than the motors.
The
The
machining.
little one is less than $50. So yeah, it just costs to machine it.
Okay, so that's
Anyway.
probably small enough to put inside the chamber in an SEM.
Yeah, it's about.
Yeah, you can put
It's
it.
the tip of my finger big.
I want to show you this machine
If
from
that's too big or not.
Kleindick.
I can send you the specs on it if you want. There's what it's called. Oops. I bought it on Amazon. It's a high tech. I have to put this on to see it. Can't read that small. Yeah, a high tech HS 35 HD.
Okay, here.
Ultra nano.
Susan, here's Yorgos's, I put it in the chat, Yorgos's contact information.
Okay, I may have it. I'll just send you the specs. I just ordered it on, like I said, on Amazon. It comes from Japan.
Okay, so Yorgos, you should be able to do it for maybe
I know. Let
with the
me
wire
show you.
going up
Yeah.
$100, $200, because you're just doing one rotation, one axis.
You know, they have also to be compatible with the vacuum, you know, and that's where we don't know what is compatible.
It's a
Try
motor.
to see if it implodes. If it's not very expensive.
You see this? I share the screen now, and
Yeah, well, that's overkill.
yeah, that must be like 7,000.
Yeah, well, that's ridiculous. It's too much.
It's
No, this looks very robust. I would try it in a vacuum.
Yeah, sure.
yeah, 2 rotational tips. Yeah, this.
You need to build it yourself, and then it'll be cheap.
Yeah, this I asked for this, and they told me 5,000. And if
Oh my goodness.
if I buy the whole thing is 7,000. I say I won't buy anything. That's it.
No, that's ridiculous. All you're trying to do is rotate a pin.
Yeah,
You don't need all the fancy stuff.
No, I can rotate through the states here because we have 5 axes, but this stage is very rough for such little dimensions, you know, and it jitters. It jitters,
Okay.
it jumps,
Yeah, but that can all be corrected by software. That's not not a big problem.
yeah, if this is corrected by software, why spending so much time to do it perfectly, you know? Because it is very time consuming for electron microscopy. One of these photos maybe 2 hours.
Okay,
or 3.
let me ask you a question. How do you rotate a needle inside an SEM? Can you show us how you do that?
There are two ways to rotate. One is, do I still share screen? I want to stop it because I'm not familiar. Stop sharing. Where is stop sharing? Anyway, there are 2 ways of rotating. Either you put
Upper right-hand corner, I think.
either you put perpendicular or horizontal. Horizontal is actually the tilt. The tilting is horizontal rotating, you know. Because the tilting in this microscope works like this, and There are buttons to do this, and some are motorized, some maybe not.
Bradley, you still there? Could you help him get his screen share off so we can tell us? I'm not sure what's going on.
Yeah. I opened.